My name is Juan. I've been a postdoc with Klaus for over 4 years. I've been part of this Pittsburgh workshop for maybe a couple iterations. Maybe this is my third or fourth. I don't actually remember. We constantly do these workshops, not only here in Pittsburgh. We have a workshop in Berkeley. We have workshops in Europe, in Jülich, in Germany, in Bremen, and we're all over the place. We think it's very important to teach people how to use our software. Hello. Okay. See if
That's the camera mic.
Oh, that's the camera. There is not a mic. Okay.
You stand here. This is.
Oh, that's the actual mic. Hello. Okay, that's better. So as I was saying, we consider it very important to train people in how to use the software that is developed in the group, as well as the different algorithms that are developed in the group. Here you can see Klaus. He is currently sick and couldn't make it here to the workshop, but he sent you his regards, and he sent you two of his best grad students, Muyun and Yi, who are very experienced and know how to run the software and will assist you in during the tutorials. So I'll be teaching Monday, Tuesday, and I'll tell you a little bit more about what I'll be talking about later on. San will be, as Yvette said, she'll be teaching, I think, Wednesday, and then Yvette and Dan will be teaching later on in the week. This you already probably know. The morning lectures will happen in this room. The afternoon labs are being held in the room next door. And then the meals are where you guys probably had breakfast this morning. So today we will be talking about, well, a little welcoming that was done by Yvette. I'll be talking about VMD. And later on, after our coffee break, I'll be talking about NAMD. We will have a little short question and answer session at noon before lunch break. And in the afternoon, we'll be doing the tutorials. And then the TAs will assist you with any questions you may have. On Tuesday, I want to talk more about like the physics behind molecular dynamics and why this is just more than making pretty movies. We'll talk about the current applications of NAMD and VMD in actual research scenarios. Also very important, we have a group picture tomorrow, so if you want to be in the picture, try to be here by noon. Again, we're going to have a tutorial session tomorrow in the afternoon from 2 to 6. You pretty much have freedom to pick any tutorial that you are interested in. And then in the afternoon, in the evening, you guys, I don't know who's presenting. Do we have volunteers for presenting tomorrow? Or
I'm not sure
we're not doing this? Okay. So then we can
maybe let me quickly mention that.
Okay.
In the past workshops, attendees were willing to share what they are doing to establish a better communication. And in response to their request, we organized a
Presentation.
workshop for participants. So you give talks, you present to each other what you'd like to talk about. Are you interested in something like that? Yes. Okay, so that is going to be done at 7.
Okay, so that we need tomorrow at 7. So
It is not for 2 hours. For each part. So who would be interested in making presentations, let's see? 1, 2, 3, 4, 5, 6. 6 people? 7? So 7, 2 hours. That would be like 10 minutes.
10
plus
minutes.
5-minute discussion per person.
okay.
And we didn't also mention, the lunches are here.
Lunch is here, yes.
But the
Dinner is on
on your
own, yeah.
And this place has many, many very nice places actually.
It-
You can walk.
Yeah. So that's yeah, you're on your own for dinner, but you can explore
So
around.
stay at 6, and it will make a lot of sense to go to a nearby
nearby place. That's Tuesday. That's tomorrow. Today, yeah, today will be done at 6. So.
But
There's the-
tomorrow at 6 is my
One question.
Yeah.
in the
In the
nearby
labs,
place
there are
and come
different
back at 7.
streams, right?
Say it again.
In the afternoon labs, there are many different streams where participants can attend. We have a choice to attend different ones.
Uh, the idea is like you attend to all of them. They're on your own, so you go at your own pace, and then you pick what you want to do. There is not gonna be like someone guiding at the front. It's like more you work on your own, and then if you have any questions, you can ask the TAs or.
So we are free to attend all of them.
Yes. Yeah. You should attend all of them. That's what is. That would be ideal. Then on Wednesday, yeah, this is Zan's program. She'll be talking about evolutionary concepts and network analysis, introduction of simulations to whole cells using her own software. It is the same format for the afternoon where you have a option of tutorials, and also her TAs will be here tomorrow. They'll be here tomorrow, but they'll be available Wednesday to answer any questions you guys may have. Then Yvette will be talking about her software, ProDie, and the A&M server. Then also talk about, we'll also have this format where you have a question and answers at noon. Again, lunch is provided here, but then you're on your own for dinner. And it's the same format. You have a series of tutorials that you're supposed to work in the afternoon.
One last thing that I forgot.
Okay.
So for the Thursday session, many of you may prefer, it's totally up to you, but if you prefer to use your own laptops as opposed to the machines that are available here, you may need to upload all the necessary software. So we will just make sure to send you all the instructions, because and on Wednesday during the hands-on session or after that, we'll make sure that everyone is ready for the next day.
Thursday.
So you will receive an email on.
So for Monday and Tuesday, you can either use your laptop or you can use the computers in the other room. So like all the software like NAMD and VMD has been installed in the computers in the next room. So you have that option. Then on Friday, Dan will be talking about his weighted ensemble.
Was the Protein Dynamics software also installed on those machines?
I don't think it was.
Okay, because I know that there's a number of requirements for that. I was just wondering. So
I.
maybe-
Your ProDie, is it installed? It's installed. Okay, so it should be there. Okay. Yeah, so ProDie should be in there then. So Dan will be talking about the weighted ensemble method. He has been developing for several years now, and the format is the same. We're gonna have a coffee break, a lunch, and then it seems Ken Dill will give an offsite seminar. Is this happening?
That's correct.
Okay.
So we thought maybe some attendees might be interested in attending that one, if they are still,
Still around. Okay. But yeah.
Usually on Friday, we, complete everything by 4. But then there's nice talk by Candy.
Okay. So a few general notes. This course is it's a volunteer effort. So like it really depends on you guys to make something out of it. The main focus are not so much the lectures, but the hands-on sessions. So the sessions in the afternoons where you actually get to do the work yourself. So in past workshops, people ask, why don't we guide people through the tutorials? And the way we think is it's better if you actually do it yourself and then you face any difficulty and then you have someone guiding you through the process rather than you're just following someone on a screen. The final, the goal of this is to get everyone here to do computational biology. I know there are different backgrounds as we hear early in the morning, but the main idea is like so everyone can do computational biology. We will provide the tutorials. We have series of printed copies that we brought with us. We can hand those for you. But also, like, all of these tutorials are available online. So if you prefer to just use the online versions, you feel free to do that. We'll provide the links in the afternoon sessions. Obviously, as I said before, the optimal course of for this to work is that people work together, you work together with the TAs, but more important is like you actually go through the tutorial and make sure that you get the answer to any questions that you may have when you go through doing the tutorials. And also, if you have your own system, if you have some previous experience, you should also feel free to play with your system rather than with the systems that are in the tutorials. So that way you can gain an extra handle on how you are going to use this new information in your particular problem. And also we're going to be handing in feedback forms so you can, after every day. So please, we ask that you please give us this feedback and return it to the TAs. This is very important to us because this way we know how we can improve the workshops for future cycles. So, I mean, Yvette already went over all the people who are going to be TAing. I'll bring your attention to the two TAs for Monday, Tuesday, Yishang and Muyuo Lihan, they're pretty well experienced. So if you have any questions, I'm pretty sure they will have answers for you. So let's move on to the content. I think I'm doing pretty well on time. So the basic idea here is to tell you a little bit about our software through examples rather than giving you like a physics lecture, which we will do tomorrow. But today we're going to introduce a motivation based on some examples. So some of you may be familiar already with VMD, which stands for Visual Molecular Dynamics. And we like to think of it as a computational microscope. And we think of it as a tool to think. And the reason this is because you're using mostly your brain through like all these visualization steps to analyze like very complex systems. VMD is freely available on the internet. You can just download it. It's available for multiple platforms, from Windows to Linux to OS X. And there are several tutorials. This is the link for the tutorials here at the bottom. There are multiple case studies. The case studies are different from the tutorials in the sense that they show exactly how a research problem was solved using these tools. So you can feel free to look at the case studies and the tutorials. Also very important is VMD can handle data from electron densities in small molecules all the way to whole cells, as you will see during this workshop. So we can do large molecules, including RNA, DNA complexes. We can do lipids, proteins, and also we can do cellular subsystems, and we'll be talking about this a little bit more as the workshop progresses. But more importantly, VMD can do all this as we like to think as a computational microscope. And as a computational microscope, what is the microscope actually made of? I mean, you know, you have a physical microscope, has lenses, has mirrors, has all this hardware. So our microscope is built from chemistry, physics, math, and obviously the software. But also we require, for many of these calculations, supercomputers, because these are computing-intensive calculations. We will be talking tomorrow more about the chemistry involved and the physics involved, and today we're going to just focus again on what can be accomplished by using the computational microscope. But what else do we have in the computational microscope? We also have a lot of graphics or geometries. We know all these systems, especially when you start looking at larger scales, the large-scale geometries start to play an important role. Similarly, we have a series of lipids. We know lipids behave very differently from proteins and from bulk solvent. And for instance, here we're going to look at this later on more in detail, how it is the coarse-grained models for lipoprotein self-assembly. We also have VMD can look at the atomic coordinates and can look how the atomic coordinates change in time, can look at volumetric data as in electronic densities, like in this case in a Buckminster fullerene. It can also link genetics, and it can also like visualize like these large complex structures that you will be seeing more during the course of the workshop. This is more related to what wet sand does. But VMD is this very capable engine that combines all these different aspects of computational biology into a single piece of software. So let me show you a little bit more of what we mean by this. So for instance, in the case of volumetric data, there is this new revolution in electron microscopy. So VMD is capable of combining not the classical look of like secondary structure of proteins and with the newer look at the electron microscopy densities. But at the same time, that volumetric density handling can also handle the electronic density around smaller molecules. And is this ability of VMD to handle the two, or like this multi-scale kind of data that make it so powerful. But we can also, VMD is also targeted towards bioinformaticians. So we can also look at not only like structural properties, but we can also look at the primary, like something like the primary structure, right, like the sequence. And you can do several things like multiple alignments, phylogenetic trees, and you cannot just do, you cannot just like look at the multiple alignments, you cannot just like do the phylogenetic trees, but you can also map them back to a structure. And then that's what makes VMD so powerful, is you're combining multiple sources of data into a single software, and this is particularly interesting in the context of biological systems. Furthermore, like when you start looking at trajectories from simulations, you can actually load these also into VMD. You can look at trajectories. You can combine those with energies. You can also visualize forces. And this is all within a single piece of software, and that's or what we call the computational microscope. And the whole idea behind VMD is that you can actually perform molecular cell biology through VMD, and that's something that we are pushing forward, and I think it's something that can be accomplished, and is based on the present success of our software. So for those of you who are not familiar with VMD, this is what it looks like. It's a very intuitive interface. So it essentially has the main menu where you can, as you load the molecules, you start to see they start showing up here. You have a TK console, which is like kind of a console where you can type commands, and then you can, if you're writing, this also allows you to develop scripts directly. And that way you're not only limited to what you can do visually, but you can also, you have a powerful scripting engine, which is based on TCL/TK. You also have the output console, which is like where VMD is going to be telling you everything that is going under the hood, everything that you may need, and to know about how a process is running. In this case, we have the graphical representations. So for each molecule, in this case, like molecule 0, you can create multiple representations, and I'll be showing this functionality later as we go through the lecture. And well, clearly the graphics window. The graphics window is where you visualize everything that you are interested in, and you can visualize multiple things in the graphics window through the graphical representations, but also you can do it through scripts, and there are other like plugins, as I will mention later on. But this is your typical VMD session, and this is what you're going to be seeing later on when we go into the VMD tutorials. So as I said before, you can also do bioinformatics within VMD. We have the sequence viewer. So for instance, if you load your own protein, in this case is ubiquitin, you can, again, here you have the main window, you have the graphics window, you have the graphical representation window. In this case, we can see that we have 3 representations here. So the first representation corresponds to the secondary structure, as you can see. And then it has one that is red, which that means that it's not being shown at the moment. It's in a licorice representation. But the bottom one is also licorice, which corresponds to this red residue being highlighted at the bottom. But what is more interesting here now is the VMD sequence window, which allows you to see the sequence of your protein of interest. And not only that, but you can, since you're also visualizing the secondary structure, you can see how different regions of the protein are mapped to the secondary structure assignments. There is also a more powerful tool, which is called Multiseq. Multiseq can do sequence alignment. In this case, for instance, you can see we have multiple. You load multiple molecules. You load, and then you can extract. Well, these are PDBs. So you can extract the sequence from those PDBs, and you can do multiple sequence alignment. You can also perform what is called structural alignment, where there is poor sequence homology, or you have gaps in the sequence, but the structures are similar. So you can also perform that kind of it's called stamp structural and sequence alignment. So that way you can not only align the structures by their coordinates, by like how they fit in space, but you can also incorporate information from the primary structure, which is the sequence. So this Multiseq is available in every version of VMD, and you can use it anytime. For instance, another interesting feature is here the graphics representation is being coloring from red to blue. You might be wondering what that is, and actually that's being colored by the charge of the residue. So what you can not only look at how the charge is, this is charged, so this is different from electrostatic potential, but it'll give you an idea. So this is showing the charge of the residues, but also you can look the charge of the residues on how that maps to the primary structure, or how that maps back to the sequence. So it also provides that capability. As I mentioned before, you can also build phylogenetic trees. So if you have multiple sequences, and you have, well, obviously some associated structures to them, you can perform multiple alignments, and then that way you can actually create a tree of how like say a series of PDBs are related to each other in this phylogenetic tree. So this is a tool that is of most use to bioinformaticians, and if you're like in early stage of building your model, this can provide some useful information. Obviously, I mean, these are tools that then you find a way how to incorporate them in your research. Yes? No.
PDB and PSF, could you just say a few things about what are the formats or something?
So that's a very technical question. So the PDB contains the sequence of amino acids and the coordinates of every atom in that amino acid, right?
Is there like a general coordinate system that's being used? Or?
So they're in reference to origin. So it's not common. So each PDB can be in a completely different space. And then the PSF has more, some more specific information, but that's like charges, masses, and also contains like the any like topological information regarding of how the bonds are and things like that. So, but the PDB itself contains like a sequence of residues, the atoms that are known, because not all of them are actually known, or some of them could be missing, or even some residues could be missing. So that's where the sequence is what is being shown here. Yeah. So I don't know if that's clear, or if anyone has any other question about how. I assume everyone knew what a PDB file was. Nope. Yes.
Yeah, I'm familiar. I don't know if everyone knows with the PSF.
Okay.
You know what it is.
Yeah, maybe we'll go more into that later. In any case, So VMD has those bioinformatic capabilities, but again, this is also a different set of capabilities that are incorporated into VMD, and is the ability to handle volumetric data. So the PDB before, and maybe this is along your question is, PDBs contain atomic coordinate discrepancy. What happens if you have a very low resolution structure, or if you have only like a blob that gives you like a probability that there's some atom in that region. So that's something similar to what you obtain from EM. And what VMD allows you is to actually visualize that volumetric data. So like if you look here we're looking at a ribosome, and this is an EM. The whole model you were looking before is an EM electron microscope, cryo-electron microscopy electron map. And what you can see here is you can, with VMD, visualize both the atomic coordinates as well as the density map or volumetric data. So volumetric data is different from a PDB in the sense that you don't have the coordinate of every particle. You just have like a well, yeah, but you can think of it as a 3D matrix, and then you have for each voxel you have an associated number with an intensity, and that is what VMD is visualizing here. So you can have time-dependent densities, like for instance for QMM calculations, like when you have an electron density. This is a time-varying electron density. So you can observe this is like effectively it's not only the coordinates, but the volumetric data is changing in time. So you can not only look at the fluctuations of the atomic coordinates, you can also look at the fluctuations of a density or volumetric data. And it's not only limited to cryo-EM data, it's also useful in the context of QM calculations, not necessarily. And then, this is a widely used tool, which is called molecular dynamics flexible fitting. That's when you fit, when you start from like crystal structures, and then you use cryo-EM data to get more realistic conformations. But we'll talk more about that later. So VMD can also be used to determine physical properties of molecules, biomolecules in particular. So for instance, you can look at hydrogen bonds, you can calculate salt bridges, you can calculate distances between residues, and you calculate for instance, surface accessible area. All of this within VMD, yes?
I'm just curious, I know that in the NAMD package that it bases the hydrogen bonds on the electrostatics.
So there are no explicit hydrogen bonds in NAMD. There are, they just show up as a consequence of the force field.
Yeah, I was just curious if there was some way to manually put in a hydrogen bond, for example, if you wanted to, that wasn't. I mean, I guess you could do that through the electrostatics.
So you can, yeah. I mean, there's a way you can add extra bonds.
I didn't know if that was like the Dahed rule or some way how people did it.
Yeah, you can add extra bonds. So you can have non-kosher terms for the force field. Yeah, you can add that. That should be in one of the tutorials. Mm-hmm.
So this is a very specific question. For example, when I'm calculating RMSD of the structure,
Mm-hmm.
let's say I've taken up the structure from the protein data bank,
Mm-hmm.
included one of the residues, and I have run the dynamics of the system and I have find the structure.
Yes.
And then I also have the structure from NMR of the mutated guy. So obviously the number of atoms and everything varies. And
Mhm.
if I want to see the RMSD between that structure and my simulated structure, I keep getting errors because of
The atom count. So you can, you can use have you tried multisig with STAMP?
No, but what I do is I manually go and then then I have to look for the atoms that are missing and whatnot.
Yeah.
I was just, if I want a quick fix answer.
Multisig.
Okay.
So you can use multisig. It'll do the alignment for you, and it'll give you the RMSD. Yeah. The RMSD she's referring to is the built-in root mean squared deviations between two atomic coordinates, atomic data set. But it works under the assumption that the two have the same number of atoms. So it doesn't do any interpolation or extrapolation. So yeah, you have to use some. Multisig doesn't care if one set has different number of atoms from the other set. But yeah, you can ask Muyun and Yi if they later on they know how to do this. Okay, so you can also calculate physical properties. This is VMD. You can visualize the hydrogen bonds directly into VMD, and you can visualize them. I'll show you guys later. And if you have a simulation, you can visualize them during the course of the simulation. Same with salt bridges, and you can also use VMD as a ruler. You can define like a distance and measure and things like this. This is particularly interesting in the context of simulations, but I'm pretty sure people find. I mean, it really depends on your problem. How do you want to use these capabilities? So you can also calculate the surface area, accessible area. You can see how much of a part of a protein is exposed to the solvent, how much is buried within the body of the protein. Also, you can calculate. We have multiple algorithms to calculate the electrostatics. So the electrostatics is now we're talking about the electrostatic potential. So you have a collection of charges. So now you can actually calculate the effective potential of those charges in a volumetric space. So this is also a capability that is available. All of this is available in the VMD you download online. So other than that, there are multiple other properties that can be calculated. For instance, even using Yvette's software like normal modes and things like this. You can measure angles, dihedrals. You can calculate RMSDs, or like root mean square differences, root mean square fluctuations. And it's usually pretty easy to calculate. So let's look at an example of trajectory analysis. So this is a 100 microsecond folding of the lambda repressor. This calculation was done actually here on Anton, in the Anton supercomputer. It was published back in 2020. I think it was published 2013. So you can see the initial conformation is an extended polymer. And actually, if you are interested in protein folding, he is an expert in protein folding. So you can ask him more about this. But the initial conformation is simply a disordered unfolded polymer. But you can see how, did it stop? Let's see if I go back. So that's the crystal structure. We start from as I said again, the unfolded, and then you can see how it collapses and then finally finds its final fold. It's obviously a very long trajectory, and this is what a typical analysis of the trajectory will look like. So you will have a number of frames loaded. You have a PSF and a PDB. Again, the PSF is required to perform the NAMD simulation, but we'll get into that later, especially when you go over the tutorials. But then it has been of representation, but here we're using a different software which is called Timeline, which is also incorporated into VMD. And what Timeline allows you is to look at particular properties during the course of the simulation. So this is clearly changing in time. There is a clear change in time.
I just had a quick question about that simulation. So that was, I'm assuming, solvated?
Yeah, fully solvated,
okay.
Yeah.
And how long did that take then?
So this probably took 2 years on Anton. Yeah, so this was 2 allocations, yeah. Maybe 3. Do you remember, Yi, how many allocations this was? This was a couple of allocations. 2 allocations. Yeah, it runs in 2 weeks, but it took 2 years of. No, it took. Well, it's hard to tell. It's hard to make a comparison of actual computer time. Like if you have a single CPU for 100 microseconds, that'll be a couple of years. On Anton, it takes 2 weeks, but they only give you a week a year. So.
Okay, so that's how it's
I don't know. Do you know, Yi? 40,000? Yeah, 40,000 atoms. So it's pretty small.
How many cores it was running?
Sorry?
How many cores it was running?
So this is a special purpose machine here in Pittsburgh. So it's only 512 cores, but they're different cores. Any case,
So, can a coarse-grained simulation be used to achieve the same result?
it's a good question. Maybe. If you tune the parameters enough, yes. It may fold the protein. It may not give you the same intermediate states, though, right? Like it will give you, you're not guaranteed to have the same. It has been sampled the same intermediate states because the coarse-grained model is going to be targeting defaulted state, not the intermediate state. So you don't know what intermediate states you're going to be missing. We're going to show an example of that in this same.
Is the timeline plugin like already installed, or is that?
It is installed,
Oh,
yeah.
okay.
So with timeline, so here you have the x-axis is time, the y-axis is whatever property you're interested in. So for instance, in this case, we're showing on the y-axis is the secondary structure. So here is very interesting because you can see like secondary structure is actually color-coded in this timeline. So you have for a turn is green. For instance, in this case, the helix is purple, like alpha helix is purple. So you can see how the alpha helix are forming, folding and folding in time. And then so you can have a very interesting look at how the folding process is happening. So you can see how for instance, this helix here is forming and things like that. So that's what is cool about timeline. We'll look more into that later. Is that you can actually look in time to a particular. You can look how a particular property that you're interested evolves in time.
It's partly useful for a long simulation to see that everything is going.
Well, it really depends what you can use it for long simulations, but you can use it for other things that, I mean, for instance, if you're looking for a cis strands or something like that,
Right.
so it will become pretty obvious when it happens. It really depends what you want to do, yeah. So it's very flexible. It's not only limited to secondary structure. I'll show that a little bit later. Right now, why don't we just play? Okay, so I'm now gonna switch to a real VMD session so you guys get a better flavor of what's going on. So this is your typical VMD. Here I'm on a Mac. And this is another folding simulation. This is a Willing headpiece simulation, folding simulation. So you can see, similar to what we saw before, it starts from a completely extended conformation. And here, where we're looking at, this is like the side chains are being colored by residue type. So the polar residue are being colored green. I don't know if it's clear to see. It looks good to me, but so polar residues are green. Let's try a different representation here. Oh, no. Let's try. So the white ones are the hydrophobic residues, and then the charged residues, like say lysines, are blue, and then the carboxylic groups are red, or anything that has a negative charge is red. And then again here we can see, we can actually start, like we are controlling, it's not a movie anymore, this is like what's happening right now. You can actually start looking at the different snapshots, and then you can, we're also looking here at the quicksort representation. It gives you an idea of how the volumetric data works. Here, those black dots are hydrogen bonds that are being formed and broken apart. You can see and you have full control over the visualization of the system. So that's a typical VMD usage, showing hydrogen bonds and it's showing a very simple representation of the side chains and showing the secondary structure being colored by, you can have multiple ways of coloring these, multiple ways of coloring, like you can assign colors based on charge, you can assign colors based on fluctuations. It's very flexible. You can do a lot of things. Okay. I don't know if anyone have any questions regarding the simulation or the visualization. This is, you can see like how falling happens. You can play it if you don't want to control it. This must have been Charm 22 Star. Yeah. This is an old simulation. Okay. Oh, okay, let's go back here. So this is a closer look to timeline. So again, in this case, we're going to be looking at the graphics window, which shows you the secondary structure or whatever representation you're interested in. In this case, we have the timeline here on the right. Again, we're looking at the secondary structure. We're looking at beta turn. Extended beta is yellow. Isolated bridge is this darker yellow. And the coils are just left out blank. So here, again, we're looking at, so can anyone guess what we're looking at without looking at the movie? What's happening? Right, like you're losing secondary structure, right? So you're unfolding, right? So, I mean, just, so you can just see that, right? From, without looking at the movie yet, you can just tell like you're losing secondary structure as the simulation progresses, right? So let's see what's actually happening in the simulation, or what is going on here. So if you notice, okay, here there's another detail here. So we're highlighting this residue here, which you can actually highlight by just selecting on the timeline window. So that's actually what's going on in the simulation is we're pulling from one end, and by doing so, we're actually inducing unfolding. And this is also something that can be, we're going to be showing later on the NAMD tutorial, on the NAMD lectures, is how to perform stir molecular dynamic simulations. But what is interesting here is like, I don't know, I mean, I want you to draw your attention again to what's going on the timeline, so that you can actually select particular residue of interest, and it will just show that residue of interest in the graphics window. Again, you can control the graphics window. You can adjust how much detail you want, like it's the kind of a zooming function. And scroll down, that's if you want more detail. That's particularly useful if you have big proteins with a lot of residues where you cannot fit all of them in a single window. Again, you can select a particular frame and a particular residue of interest. So it gives you a flexibility of looking at what you might consider is interesting during your simulation. Again, you can control how the simulation goes back and forth, and you can select different residues that you might consider are of interest. Again, this timeline is included in every, it's included with VMD. If when you download it, you don't have to install anything. Yes?
When you play the simulation backwards, is it the same as the protein folding? Or
yeah, that's a very good question. It should be, I think I talk about that in the next lecture. Yes, if it is, the idea when you do unfolding in this way, by pulling apart, is that the degrees of freedom that are perpendicular to where you're applying the force should be in quasi-equilibrium. So in a way, you could argue that you could reconstruct to some extent the folding pathway. Yeah. A folding pathway.
One question. Why does the colors not change when simulation is totally unfolded?
On the left. So there's a, by default, VMD only calculates the secondary structure for one frame and then applies that to all other frames. So he doesn't, by default, he doesn't update he doesn't recalculate the secondary structure for every frame. There is a way you can do that. You can tell him to recalculate the secondary structure for every time step. It becomes a little bit sluggish, but you can do it. By default, he assumes that you're usually sampling near a like a folded state, and as such, the conformation is not going to change much. But if you know that it's going to change, then you can tell him, okay, I want you to re-update the secondary structure every frame. But in this case, that was not the case when the movie was made. So that's why it looks like that. So, well, that's an example of only one of the many plugins. VMD has several built-in plugins. All the plugins are available on this URL here. So, for instance, you have modeling plugins. You have, like, for instance, the auto-ionize, auto-PSF that allow you to ionize your simulations, allow you to automatically generate your PSF for the simulations. You also have things like the FFTK, which allow you, for force field, the force field toolkit, which helps you with parameterizing if you have a novel compound or if you have a novel residue that you want to incorporate in your simulations and is not available as part of the standard force fields. There are several analysis plugins. Some of you may be familiar with APVS, Poisson-Boltzmann electrostatics. So there is an easy way to do APVS directly from VMD. The normal mode wizard, which is developed by Yvette Bahar and links directly to ProDye. And maybe, I think she talks about that in her lecture, and you have a chance to play with that during the tutorials. There is their RMSD tool that allow you to calculate root mean square deviations. There are the SolBridges plugins that allow you to calculate SolBridges as the simulation progress or for your structure of interest. Timeline, we show briefly right here. There is another series of visualization plugins that are more targeted towards like how do you actually present your science. For instance, there are ways that you can actually calculate the dipoles in your protein, and obviously you can do this as the simulation progress. There are multiple other tools like a view change renderer that we use a lot. That is really good for making movies, allows you to change the viewpoints and generate transitions between different viewpoints. There is also like the simulation plugins. We're going to be, he will be presenting later a new plugin that was developed in the group. It's called QuickMD, that is supposed to help people who have little experience with molecular dynamics to set up their simulations more quickly. But we have the interactive molecular dynamics, which allows you to directly interact with your protein, with your simulation as it goes. So this interactive molecular dynamics connects NAMD and VMD. So as your molecular dynamic simulation is ongoing, you can say if you want to rotate a bond or you want to do interactively alter the course of the simulation, you can do so. There are also a series of data plugins. Yes?
With what you were just talking about, could you do SMD with that?
Yes, you could do SMD. Yep. There's a tutorial that teaches you how to do SMD with through IMD. I don't know if we'd include it, but if you want to play with it, I mean, we all have experience with IMD, so you can do it during the tutorials. So the data plugins are different. Well, there are a series of plugins that allow to import and export data from VMD into other more foreign platforms. And well, as you can say, there are several multiple plugins, and it's just they just keep growing because there are several of them are developed in our group, but many of them are also developed by other people outside of our group. So VMD can be thought of as like a kind of a two-phase program. It has a very powerful compute engine, and the compute engine is a very important is a very important component of VMD because, as I mentioned before, it allows you to calculate electrostatic fields, and this is very important when you are building your simulation. For instance, when you have very charged systems like RNA protein complexes or viral capsids, the ion placement is very important. And the way that we normally do the ion placement is by calculating the electrostatic potential of the protein and then placing ions in such a, placing the ion with the right charge, either positive or negative, at local minimum of the electrostatic potential. And this is something that belongs to this compute engine part of VMD. And that's also accelerated by the use of GPUs, or graphical processing units. I guess they're more common now than they were 5 years, 10 years ago. But the idea is like you can buy one of these accelerators and you can do, for instance, electrostatic field calculations and ion placement calculations 20 to 40 times faster versus a single CPU doing the calculation. Also, like for molecular orbitals, it's also optimized using GPUs. The acceleration is 120 times faster, and then that allows, like as I showed earlier, the movie where you can actually see the molecular orbitals changing in time. There is another very interesting part of VMD that is also accelerated through GPUs, which is for calculating something that we call implicit ligand sampling, or also like calculating like essentially a three-dimensional potential of mean force, which is simply this is simply calculated by essentially building a volumetric histogram and counting the occurrences of observing some particle within a voxel, and that way you can actually calculate pretty accurately free energies of binding of say like in this case CO and oxygen in a protein. And this, as you can see here, like these are channels. So here you have a protein, but then these lines are representing channels through and through these channels, oxygen and CO are permeating inside of the protein. And you can do these kind of calculations from your simulation, and the analysis is done in VMD, and the fact that you use a GPU is going to give you a 20 to 30 times the speed up, depending on the GPU you have, and obviously the simulation you have. But the message here is like VMD is not only making pretty pictures, it's not only using the TCL scripts, there's also like hard-coded subroutines that use the GPU acceleration. The electrostatics here, this can be observed a little bit in terms of a grid and how, like a little bit of the details, like you have the protein in the grid, and then you assign charges to the grid, and then you calculate the effective electrostatic potential at every point in that grid, and that calculation is what is being optimized in the GPU.
Is there a particular plugin that
You can use BallMap for that. I don't think there's a tutorial for that, but there are at least 3 papers on doing that.
WAM and some other.
Well, yeah, this is simpler than WAM because you have only 1 degree of freedom.
Okay.
And this also requires that your simulation is well sampled. So, I mean, there are some caveats to it, but you can do it, you know.
Okay.
I have a question.
Mm-hmm.
So how is the mark calculated? I mean, does VMG do something?
How is what?
How is the molecular or
So these are from Gaussian.
Ah, okay.
Yeah. So you can load your cube files from Gaussian into VMD. Yep. Yes.
Is there some guidance on compiling in order to take advantage of the GPU?
So we provide the precompiled GPU versions. So the moment you download it, you should have there are options to download the GPU accelerated version, compiled versions of it, and it includes as long as you have the driver, you shouldn't have to recompile.
Okay, I'll ask more later.
Yeah, yeah, but it's yeah. But, I mean, compiling you can always try. Yeah, compiling VMD is kind of an endeavor, but yeah, you can do it. But then, okay, so the other side of VMD is what Klaus likes to call the artist, and it's because you can make really pretty images. I mean, you guys are probably familiar with this one, which this was the cover of Nature. We made this for our paper in 2013. But I mean, we have an entire tutorial that teaches you how to do pretty images. This is our chromatophore. I'll be showing a little bit more later on. This is a ribosome, and then you can see here is with a sec. I think this is sec-Y, and this is a nanodisk. So this is a work done by J.C. Gumbert, who is now a professor at Georgia Tech. He was a Klaus's postdoc and a grad student postdoc, and it's a very pretty image made by Bo Liu, who is a grad student in our lab. But and this is the mechanosensor channel, and you can see this is the membrane, and you can see diffusion of ions. But VMD has this ability of making really pretty pictures, and as I said before, there's an entire tutorial devoted. One of my colleagues, Bunshong Go, he developed. There's a YouTube video. You can go and look, and he teaches you how a few tricks that they're in the tutorial, but some people bypass them. So sometimes if you go to the YouTube video, you can get a better idea of what you should do to make your pictures look prettier than what they currently are. And obviously, you can also make movies. This is a completely VMD render movie. So this is a 20 million atom simulation. You can see how like the little fluctuations are not the projector. They're actually the simulation. It contains lipids. Everything here around here are lipids. You can look at this whole movie was rendered using VMD, so you can actually control the transparency of the surfaces. You can control the lights. It's like a really powerful, almost like painting tool, right? Like it really depends on what you want to do and how do you want to accomplish it. So for instance, here where you can see this lighting effect is also accomplished with VMD. It's different than adding a light on Photoshop, because you're not gonna cast a shadow, like a three-dimensional shadow, if you do such things in Photoshop. Here, this one also has a very nice lighting effect at the bottom. So these shadows here are very realistic, because we use a very sophisticated ray tracer that was actually developed by one of the programmers in the group. But the lights are very, very, very important to a lot of these images. This one here, for instance, you can see the cat. I believe this is a graphene. I'm not misremembering. It's a DNA translocation. And then you can see how the shadows are also like three-dimensional shadows, unlike something you will get from like trying to do this on Photoshop. So let me show you an aesthetically pleasant presentation directly of, this is VMD running on my computer. So you guys probably don't need to see this. So again, you have all your molecules loaded here. Let's just ignore that. So this is actually cryo-ET from a cell. So you can see the nuclei. So the nuclei, you can see the cell membrane. And this is one of Klaus. Klaus has been working on this for something like 3 years. So it's a shame he's not here to tell you the whole story. But I'll do my best. So that's, he's been working on this little part here, which is a chromatophore. This little system here, when fully solvated, and atomic resolution is a 100 million atom system. You can, I mean, this is running on my computer. It's not a movie. So if you have it's a laptop. So if you have a more powerful, it's actually draining the battery. But if you have a more powerful computer, you can imagine how much better it will look. So here, this is the chromatophore. These are ATPases. But let's look more closely. These are the light harvesting proteins, the light harvesting complex 2. We're going to zoom in. Again, this is all running live right now. And it contains everything inside, the quinones, and all now rotating around. It's looking very pretty. But I mean, the message here is like anyone can do this, given you have the amount of time to do it. Let's, so yeah, there's nothing special with my version of VMD. It's just the one you download from the web. So we have, I got rid of the depth crewing there. So you have those nice transitions where you're changing the transparency of the surfaces. And again, all of this is this is real time visualization. So this was so like everything that I've been showing has been 1 or 2 papers from Klaus. So like for instance, this curvature of the light harvesting complex 1 was a big debate back in the day whether it was flat or was curved. So every single of these images actually contains a lot of science. It's not only pretty, but also contains a lot of science. But the message here is that you can also make your science look pretty. And you can actually render a high-quality movie. This is a low-quality movie. I'll show you a high-quality movie in a second. Again, the model contains, every atom contains every prosthetic group. And let's look at the ATP synthase. And again, like this is only a small part, well, of a whole cell, but it's not as small as a single protein. It's already a subcellular component. Okay, so let's talk more. Have a few more minutes. Okay, let's talk about another series of plugins available in BMD. So we have the network view plugin, which actually allows you to map allosteric pathways on proteins. The network view, there's a tutorial if you're interested in this. We're familiar with it, so if you want to do the network analysis tutorial, you're welcome to do so. But I mean, if you haven't done NAMD VMD, I suggest you first do the NAMD VMD tutorials. And then if you're interested in more advanced, this is a really good one because it allows you to map allosteric pathways on a protein. As in this case, you can see it's showing like how two distinct parts of the protein are connected. There is also the force field toolkit, which assists in the parameter development, charm parameter development, and that's important. It's only for charm compatible force fields at the moment. There are efforts to try to extend it to other force fields, but if this is particularly useful if you have a drug that you want to see how it interacts with your protein of interest, and there are no parameters for that drug, you can parameterize using the force field toolkit. If you have some unusual residue or some post-translationally modified residue that you want to see how it affects the dynamics or the structure of your protein, you can parameterize it using the force field toolkit. There are a series of other toolkits. There are other interesting plugins, the normal mode wizard, which, as I mentioned before, you will be hearing from Yvette a lot more. The PropKA interface, which is, it's actually, I consider it to be very important because when you're building your system, the protonation states of titratable groups are set to a default value, which might not be the value of interest for you. So PropKA allows you to calculate the right protonation states of your titratable groups. And then, there are a series of other plugins that were released later. We have, as I mentioned before, we have a very powerful ray tracer, the Tachyon ray tracer, which is developed in the group. We have a novel ray tracer that is not mentioned in these slides, which is the optics ray tracer, which allows you to, it's not much power, it's a much faster GPU-dependent ray tracer. And I don't know if we have GPUs there, but if I may be able to give you guys a demo later on. But the optics ray tracer is it's a new feature in VMD, and you should, if you have a GPU, if you have a Mac, you can look at it. It looks very nice. We have multiple updates on the molecular dynamics flexible fitting set of tools. We have the XMDFF for low-rate crystallography. We have a new MDFF GUI for those of you who are interested in combining electron microscopy into your simulations. There is a complete GUI that prepared the simulation and guides you. We have a tutorial, but now you have a whole, like an entire GUI dedicated to preparing these kind of simulations. One that is, I mentioned before, the force field toolkit. So you have a new parameter. This is what the new force field toolkit looks like. You have your molecule of interest. He makes sure that you have the waters located. I mean, charm is normally parameterized in the context of water. So it makes sure that you have your waters well optimized and your geometries are well optimized, and it helps you with finding the dihedral scans. And I mean, molecule parameterization is kind of a tedious process, so it assists you going through that. We have incorporated inside of a timeline MDFF analysis. A very common feature in MDFF is you want to see, you have your electron microscopy data map, and you see how well your structure fits into it. So you can see, for instance, like regions that are not well correlated are going to be red, but you can see how the MDFF is progressing as it makes the secondary structure fits better into the density. So you can actually combine timeline with MDFF, and you can see how your local cross-correlation is evolving along the simulation, how well the protein fits into the density. So very important is that our software, VMD and NAMD, work together quite tightly. I don't know how many people here use supercomputers, but NAMD is widely installed in pretty much every supercomputer in the US. And VMD is also installed in many of them. I mean, it's not that known that VMD can also use a supercomputer for multiple analysis, but you can. It actually supports MPI and some other. I mean, it allows you to write your own parallel script. If you have large data sets, which is particularly useful when you have large systems. VMD is widely used. I think this is really not updated. We have over 10,000 citations right now. It runs on multiple operating systems. It runs from a laptop to a desktop to a supercomputer. There is a mailing list. If you guys are interested, it's very active. If you have any question, you just send it to the mailing list, and usually you get an answer pretty quickly. There is the tutorials, the mini tutorials. Everything is available online, and it's free for everyone. There are user surveys that we ask people what they would like to see in future releases of VMD, or if they have any requests, or what if they think we're lacking in some area. The tutorials, as I mentioned before. So we go from the very basic how to use VMD to how to make movies and how to like check your structure, to like how to visualize your QM calculations. And well, so VMD is the effort of a lot of people. It's as Yvette mentioned before, this is a big NIH center. We are NIH. The development of VMD and NAMD is supported by NIH. The main developer is John Stone, and but the reality is like there is a lot of people that contributes different plugins and contribute different test case and contribute like either like tutorials or tutorials, mini tutorials, or documentation, or assisting in the forums and mailing lists. I think that's it for this part. I think we have a questions and answer session right now. So if you guys have any questions for lunch break, before coffee break, yes?
So this is a trivial question. How do we set that viewing and orthographic by default every time we open the VMD?
How do you set it to orthographic by default? So there are multiple ways. There is a VMDRC file, .vmdrc, in your home directory, so you can change that. There is an extension. Oof, this one is very slow. Oh no, it's not that it's slow. There is an extension that is called, it's not loading for some reason here. It's called VMD Preferences. Here it is. So if you go to extensions, and then at the bottom it says VMD Preferences. You can come to display, projection type, and then just change it to orthographic, and then you can just save it, write settings to VMDRC, and he'll update your VMDRC by default. If there are any other defaults that you want to change, you can do it on the VMD preference setup, yeah, but you can, that's where you can change it more directly.
Can you talk me a little bit more about the PropK or the ProPK?
PropK.
How do you assign the protonation states and what do you mean when you say correct protonation states?
So for every pH, you're going to have different protonation states, right?
Sure.
So, and it's also going to depend on whether or not you have if it is exposed or not.
Sure.
So this is based on entirely based on electrostatics. This is running, or I mean, it depends on Prop KA, right?
Yeah.
So that's a program developed by Nathan Baker.
Okay.
And so what it does is it takes a PDB and it estimates what the protonation state, it assigns hydrogens to the, like say, if the histidine delta has to be protonated, it puts a hydrogen there and so on.
Yeah, I understand that, but my question was, what is it based on? Is it the discrete protonation state or predefined? They make some educated guess. I'm looking for how they decide. What's the model that they use to assign?
So the model they use is very similar to how our ionize works. And is you calculate an electrostatic potential and then you start querying, you put a hydrogen there, right? Yeah. And then you put a hydrogen there and you recalculate the electrostatic potential. And then if it improves, then you keep it. If not, you don't keep it. So it's kind of a Metropolis way you're going. Mm-hmm. There are multiple ways.
Yeah, I'm currently doing rapid exchange.
But that's for constant pH, right? Yeah. So, but that's a different problem. Yeah. So because so yeah, that's a different problem.
I was just curious though. I mean, not all the parameters would be available in the standard Charm 22.
Mm-hmm.
States that are protonated, I guess to be charge neutral, if you want to call it that. I mean, for example, arginine is usually included. I know in Charm 31 they have some of those. I didn't know whether I guess NAD has been updated yet for more current Charm releases, because I thought it was up to like Charm 29 or something.
It has been up to 36. VMD was recently updated, like the auto salvation tool and the auto PSF tool.
Well, I mean in terms of topology file.
Yeah, that was updated.
Okay.
So, but then if you, there should, I mean, if
I didn't
for,
know all the amino acids whether or not you actually had the two available protonation states.
well, I mean, if it is part of CG and FF or Charm, you can use it. If it is not part of that, you'll have to parameterize it. Yeah.
Right.
Any other question? Okay, we can have a coffee break then.